Hello everybody. Now let us continue further discussion on the topic of micellar formation. So in the previous lecture we were looking at formation of micelles and formation of different kind of architectures involving the existence of amphiphiles. Now we are going to go on to the next topic where we are going to look at the supramolecular association of individual amphiphiles which in a resulting way form a micelle which exhibits micelle like properties. So now we are not really worried about a particular molecule, a covalently bonded backbone which has got say polar heads, non-polar head. We are not going to talk about such specific amphiphiles, but now we are going to talk of an amphiphile which actually supramolecularly interacts with another amphiphile, and then this resultant structure exhibits micelle formation or vesicle formation and so on and so forth. So now we are going to look at supramolecular amphiphiles, and we are going to look at the resulting structures and topological connectivity of supramolecular amphiphiles. I have designated it as SA, and we have already seen that the conventional amphiphiles have got very complex aggregation pathways, and the same thing also holds for supramolecular amphiphiles. And there is a very big library of both small molecules as well as polymeric supramolecular amphiphiles which are actually connected by NCIs, which I call as non-covalent interactions. So now let us look at the different types of supramolecular amphiphiles. Number one is the side chain amphiphile, in which there exists a hydrogen bond between the hydrophobic and hydrophilic portions of the amphiphiles. So let us now consider, for example, we have got and there is a donor here, and here we have got an acceptor. Okay, so this is the acceptor A, and this is the hydrophobic end, this is the hydrophilic end of the amphiphile, and now there exists hydrogen bonding between the donor and the acceptor, and the donor comes from the hydrophobic portion of the amphiphile, and the interaction is with the acceptor, which is the hydrophilic portion of the amphiphile, giving rise to this side chain amphiphile. So this is the first classification of supramolecular amphiphile. The second one is the double chain amphiphile, where we have got an amphiphile which has got a donor region and there is an acceptor region here A, but you see it has got a double chain and there are now non-covalent interactions between the donor and the acceptor regions, that is there exist supramolecular interactions between the head and tail regions of the amphiphile. So, this is the head, this is the tail, this is the tail, this is the head. So, there exist now supramolecular interactions between the head and the tail regions of the amphiphiles. The third one is referred to as a Bola form. The Bola form has got two hydrophilic head groups at the end of the hydrocarbon chain. So, if you look at the covalent connectivity only, then it refers to the presence of two head groups, and these are connected by a hydrophobic chain. So, this constitutes a Bola form. So, we have got these two hydrophobic head groups, hydrophilic head groups which are connected by a hydrophobic alkyl chain. We also have the corresponding supramolecular equivalent of it, where we can have the donor and the acceptor interacting via specific NCIs to give you the Bola form. And when such an association takes place, these are referred to as Bola amphiphiles. The Bola amphiphiles, they constitute a supramolecular linking arrangement, a supramolecular connectivity. And so, this is referred to as a Bola amphiphile, where you have got supramolecular connectivity and the two amphiphiles are connected by supramolecular linking arrangement. For example, we can have, this is the polar part, we have the acceptor, we have the donor, and then we have another donor compartment, and this is the polar part, and there are specific interactions between the donor and the acceptor to give you the Bola amphiphile. The fifth one is referred to as the Gemini amphiphile, where we have got two surfactant molecules that are linked at their charged head group. So, the head is actually charged, and the surfactant molecules, they are linked at the charged head group. So, for example, let us take this particular amphiphile. This is positively charged, this is negatively charged. So, there exist electrostatic interactions between these two surfactant molecules at the charged head groups. They are referred to as Gemini amphiphiles. We can have another arrangement where we can have the positive charge and then there is a negative charge which kind of insulates the positive charges, and this gives rise to a Gemini amphiphile. And in all these cases of supramolecular association, it is the hydrogen bonding or the electrostatic interactions or charge transfer interactions or halogen bonding which stabilizes this supramolecular assembly. And these supramolecular amphiphiles are of a highly tunable nature, and they are also and you can also reverse this phenomena of association because of the dynamic nature of NCIs. So, the supramolecular amphiphile formation is both dynamic and it is of a reversible nature, keeping in mind that the interactions which are involved are the non-covalent interactions. So, with this background of supramolecular amphiphiles, let us take up some specific examples representing these supramolecular amphiphiles. For example, we will look at a complex which is formed between melamine plus the quaternary salt of isocyanuric acid, and this particular supramolecular amphiphile was designed by T. Kunitake at Kyushu University. So, let us look at the structure of this particular supramolecular amphiphile. There are the amino groups here, and we can have now and this is now connected to this is a quaternary ammonium salt. So, now you can see the extensive hydrogen bonding which exists in this particular region, which is possible by NHO and one NHN hydrogen bonds. So, there is this particular supramolecular amphiphile, it forms transparent dispersion in water and gives a disc like aggregate. So, you have a disc like aggregate and forms ordered bilayer structure. Now, let us go to the next example. In the next example, we will now look at the formation of a Bola amphiphile. So, we are starting here with plus in the vicinity of this aldehyde group, we now bring in a amine group So, you see that the ends are positively charged, and if it were to be a pure covalent backbone, that will be a conventional Bola amphiphile, but now that is what we are going to do, that we are now going to form the covalent bond, and this depends upon the pH of the solution. Under pH controlled conditions, that is more under basic conditions, it will form the corresponding E mean, and we have the positive charge here, and again here we are going to have the so this gives rise to a covalent bonded Bola amphiphile. Okay. And under base acidic conditions, we know that the imine bond is very sensitive to pH, and if you lower the pH of the solution, make it acidic, then it will again go back to the starting materials where due to the cleavage of the CN bond, due to the breakage of the CN bond. And this particular property where the CN bond formation is controlled by the pH can be used to deliver a drug, for example, in cells which thrive in an acidic environment, for example cancer cells. So, in case of cancer cells, the drug release at low pH can be performed, and this Bola amphiphile has been known to form micelles in water. So, it forms the micelles in water, and these micelles can now carry the drug molecule, because the drug molecule will have hydrophobic portions. So, the hydrophobic portions interact with the micelles, and then the drug is being transported, and wherever the conditions are low pH, that means acidic condition, it breaks this particular CN bond and the drug can be released. So, you have the micelle and then you have the drug impregnated, the CN bond breaks, the micelles are disrupted and the drug can be released at the site of action. So, depending upon the pH, we can control the CN bond formation, and that can be utilized in drug delivery processes. The third example we would like to take is of a Gemini amphiphile. Where we consider (13 carbon atoms), and so we consider this particular Gemini amphiphile, which has now got the positively charged head groups which are connected by a covalent backbone, and this particular entity forms foams. So, if you take it in a vial, there will be in water, this will form foams. So, it will be all white foams that it forms, and in the presence of QQ bit 7 urils, which is actually a cylindrical kind of tub shaped molecule. What these cucurbitals does is they separate the hydrophobic chains from each other to form these kind of entities. So, the interactions are prevented. So, this particular aggregation which between the side chains, which was giving it the desired stacking property, is now prevented. These side chain interactions don't take place, and this increases the solubility of the amphiphile and prevents foaming. So now you get in this container transparent aqueous solution of the Gemini amphiphile. So now the solution becomes transparent, and then you can actually reverse the process. Again, you can actually again disengage these cucurbitacil 7 urals by the addition of 1-adamantane hydrochloride. So if you add this particular compound, then it will form again the starting material, which again starts forming. So the addition of this 1-adamantane hydrochloride is now able to again capture these cucurbitacils very efficiently and then release this Gemini amphiphile, which again starts forming foams. And this particular Gemini amphiphile was developed by Zhang from Tsinghua University in China. And another last example I would like to discuss here is the formation of reverse vesicles using nitrogen bonding. And here we would like to see, we have now a antimony 3 salt and it forms so 1, 2, 3, 4, 5, 6, 7, 8. So there are 9 carbon atoms. So we have 9 carbon atoms, and this particular surfactant forms the reverse vesicles, where we have got the polar parts sticking to each other and the non-polar parts inside, and again the non-polar parts on the outer side. So we have the non-polar part, the polar, and then normally we have the non-polar and the polar in a typical vesicle. Here we again have the polar part sticking to the interior and the non-polar part again protruding out into the interior. So the exterior and then the interior have the non-polar hydrophobic part. So this is referred to as a reverse vesicle. Reverse vesicle. So via this nitrogen bonding contacts, it is able to form the reverse vesicles. And how does this happen? So we have the non-polar part and we have the antimony, and then the antimony is able to engage. We have another antimony here, and so this is able to engage in short contacts via this. Similarly, this antimony is also able to engage in short contacts from and then this will continue here. So this again we can have short contacts. Okay, so we can have, so this kind of nitrogen bonding contacts can lead to the formation of these reverse micelles. And then we have already learnt in the previous lecture that sonication can lead to the formation of these SUVs, which are the short unilaminar vesicles, which are actually the analogues of inverse biological cell. These are analogues of inverse biological cells. So, we looked at in the previous lecture, these small unilaminar vesicles can be formed by the process of sonication. So, here also we can do the process of sonication and we can generate the inverse biological cells. So, with this background, now we can look at some of the important applications of these amphiphiles. And in this regard, it is important to first prepare the Langmuir-Blodgett film, which contains the monolayer formation, and then on this monolayer you can now put the next layer, which will form the bilayer, and in this way you can actually increase the orientation of these surfactant like molecules at the water interface. So, how do we do that? So, we essentially have now what we take is a trough of water. So, we have the water molecules here, we put in a substrate, and we will see that the molecules, the surfactant molecules, we will start sticking to the surface. So, now you have this is a solid surface, this is the monolayer, this is the monolayer, and at this point you will see that you will have the surfactant, the water and the solid phases. So, this is called a 3-phase contact, and now this is being pulled at a certain rate. This particular solid is being pulled at a certain rate. So, when it is being pulled, the surfactant molecules now start forming the monolayer on top of this particular solid surface. The solid surface, it can be made of silicon, other metals or metal oxides. So, they are polar, they have electropositive character, and therefore, they can interact with the polar regions of the amphiphiles or the surfactant molecules. And this is the aqueous layer, which has been confined to this particular trough. And now, once you have formed the first monolayer, then you can again bring this monolayer in contact with this trough, which contains the surfactant molecules, and now the hydrophobic parts will stick to this particular monolayer and form the bilayer. So, in this way you can create you can actually create a new monolayer, and this leads to the formation of a bilayer coating, and in this regard you will be able to create highly ordered structures. And the construction of such highly ordered structures, where you are controlling the degree of orientation via molecular tuning of the hydrophobic chains. This particular technique was developed by scientists, that is Langmuir and Catherine Blodgett, and therefore, this process is referred to as Langmuir-Blodgett film. So, this Langmuir-Blodgett film has now been applied to understand different interesting processes which now happen at the interface. So, because these processes happen at the interface and they involve the presence of self-assembled monolayers, we refer to these as surface self-assembled monolayers, that is we call these as SAMs. And this surface SAMs, which contains a densely packed hydrophobic chains, and these also constitute a ordered structure, can now be utilized to do some interesting chemistry at the interface, and compare these interfacial processes and the rates, for example, of different reactions at the interface in comparison to a process which is happening in the bulk. And in order to understand the effect of the interface on a particular property, or for example any chemical reactivity of a particular chemical reaction, now these interfaces have been extensively utilized to understand different processes happening at the interface. And this field was first developed in 1989 by George Whitesides. And we have already heard about some of the most important applications where we have assembled thiol molecules containing sulfur on the gold surface. Because of the favorable gold sulfur bonds, these monolayers are able to align themselves on the gold surface, and then different chemical processes can be investigated. Now, this the thickness of the layer which is formed is equal to one molecule thickness, and this thickness depends upon the layer thickness depends upon the length of the molecule and the contact angle the axis makes with the surface. So, let us see this. So, we have now got a gold surface, we have got now the sulfur containing so it depends upon now this is the layer thickness D, and this is the contact angle it makes with the surface. And normally the strength of these gold sulfur interactions are comparable to that of hydrogen bonds, and therefore these thiol molecules are able to assemble in a very clean manner on these gold surfaces. Now, when this process of self assembly takes place, there are some interesting steps. This does not happen in one step. There are interesting sequence of steps which takes place. Let us look at those two steps. Number one, the first step is the fast step. The first step is the fast step, I am putting it in quotes, and takes minutes to achieve 80 to 90% thickness. To achieve 80 to 90% thickness, as well as the magnitude of the contact angle. This is the first step. So, this is a diffusion controlled step. And in the second step, which is a very slow step, a relatively slow step, it takes hours during which the final thickness and contact angle are reached. That is, it is only after a period of say at least 24 to 48 hours, 24 at least 1 to 2 days, that this process is complete, when the final value of the thickness, or you can say the converged value of the thickness and the contact angle will be reached, such that this involves the process of ordering and packing of the hydrophobic chain into a clean self assembled monolayer on the surface. And when these steps are taking place, what is happening is that also at the molecular level, Why is the first step very fast? Because it involves the formation of large number of these gold sulfur bonds. So, the formation of this large number of these stabilizing gold sulfur bonds makes the process of coverage 80 to 90% complete. So, this is the interaction between the sulfur with the gold interface, and in the second step what happened, it is actually the side chain interactions, the interactions between the hydrophobic chains that need to accommodate all possible favorable contacts, so that we have a tightly packed structure and overall dense and firm packing. So, it is the interactions between the organic chains, the side chain interactions that occurs in the second step. So, this leads to the creation of a very favorable surface, and now these kind of surfaces have been prepared and different kind of applications have been performed. For example, people have looked at modified thiolate units, the way they have put different kind of functional groups, for example, a redox group, and then you can study the redox property between the gold surface and the receptor, which is a redox unit, and you can also put different kind of donor atoms on this. You can put different kind of donor atoms on this hydrophobic chain and then study different kind of binding affinities of anions, etc., and a whole new chemistry can be performed. And just to appreciate this particular aspect, one last example I would like to tell you. So, we can now say, for example, we have got a ferrocene unit. We have now got a ferrocene unit and we now connected with NH, and here we connected with CO. So, this is connected, okay, basically this is connected to the surface here, and now we can actually have the ferrocene unit contains iron. So, we can now study the process of electron transfer or the redox process from the ferrocene to the gold surface, and we now put in an anion. When we now put it an anion, the binding of the anion takes place and the affinity is increased. The affinity is increased. So, why is this affinity increased? Because if you were to consider the binding in the absence of the gold substrate, then you see that this these sulfur containing rings, which are called as which are actually present here, they are free to have conformational flexibility, they move a lot, and therefore the anion binding processes are weak. But now when you have assembled this particular unit on this gold surface, then this ordering of this ligand leads to enhanced anion binding efficiency by several orders of magnitude in comparison to those receptors when you do not have the gold surface. And now you can actually study the redox process, which will be different in the presence of anions compared to the case when you do not have the anions. So, redox processes, anion binding affinities, and enhancement of these anion binding affinities when you actually template the ligand on the gold surface forms these favorable gold silver contacts, and therefore results in pre-organization of this particular ligand by the modification of the surface to have enhanced binding affinities. So, this is one very interesting application of the self-assembled monolayers. So, with this we come to close we can now close the discussion on this particular topic. In the next lectures, we will now go on to another fascinating class of molecules which are called liquid crystals, and will further look at their characteristics and properties. Thank you.